A very warm, a very warm good morning to all of you. So today I am pleased to introduce Sanjay J. Jambhulkar sir. He is head Nuclear Agriculture and Biotechnology Division, BRC Mumbai. He is alumni of IRI. He did his MSc and PhD from this premier institute itself. He joined BARC in 1991 and he has a very vast experience of working with development of mutants, especially in crops like sunflower, brassica, and then other wheat and other cereal crops. He won various awards at different arena. He is an illustrious scientist having a big list of awards from international and national institutes and society. He has published more than 100 research papers in different area of mutation biology. He has very good publications, especially in the area of novel mutation identification in sunflower, Indian mustard, and rice. And he developed many improved genotypes also using the tool of mutation. So with this brief note, I request my coordinator that to welcome today's invitee speaker. So today, sir,
Yeah.
So today, sir, is going to give a talk on induced mutagenesis for crop improvement. So now I request, sir, to please continue.
So good morning, everybody, and thank you very much to Dr. Ranjit Ranjan and his team to invite me to deliver lecture on the mutation breeding. So I will start my lecture. Is it audible there? Yeah. So Bhabha Atomic Research Center, it's a premier institute in the field of nuclear sciences in India and in the world. And we deal with the use of radiations and the radioisotopes in the various aspects, like industry, medical, and even the national security and other things. And one of the major application is in the agriculture also. So we try to prove that generally the people know that the nuclear energy is for the some kind of the devastations and all these things. But there is a lot of applications that we say as a peaceful use of the nuclear energy. And in one of the area is agriculture. So we work on the nuclear agriculture and biotechnology division for the crop improvement program. That is a major one. But beside that, we have the application in the soil science, application in the entomology, application in the tissue culture, and even the food preservations. But this part I am not going to deal with. I will try to concentrate only on the mutation breeding. Hopefully, my time is, Ranjit, 1 hour? Okay. So I will try to complete. What I will try to do, I will try to give you the example rather than going to the details of the techniques, because you know the techniques even up to the genomics. Okay. So I will not go into the basic of that, but whatever we have done, whatever world has done, so that I will try to give the example. This is the all of you are well aware that our present yield is a 282 million tons, and what we need is in the future it's a very high demand, that is 333 million tons. Whatever we got so far, it's mainly it is a product of the Green Revolution, and the Green Revolution happened due to the dwarfing genes, and Dr. Swaminathan is a father of Green Revolution in India, and he got that World Food Prize. But the reason for that is a isolation of the natural dwarfing mutants. And that is called as the spontaneous mutations. That is SD1 in the rice and RST1 and RST2 in a wheat. These are the spontaneous mutations. And in the world has got the whatever production has increased in the world and India, it is basically because of these genes. This germplasm was used in the crossing program. The world has used this, and whatever varieties are there, it is out of that. So suppose I say we are getting the food due to the mutations. Had these mutations would have not been isolated in the nature, probably would have not got this kind of the jump in the yield. So if I say that we are living or we are getting the food because of the mutations, so hopefully I am not wrong. And I feel that you will be also agree with me. So that is the magic of the mutation. That is the magic of the mutations. And the mutation, all of us, all living organisms on this globe, it is evolved through the mutations, whether it's animals, plants, microbes, and that evolution is still going on. But we do not know what kind of the mutations are occurring with me or in the human being or in the plants. So evolution is mainly due to the mutation, and in the plants, then it is a recombination. That is what the magic of the mutations. This is the, though we say that we are self-sufficient in the food grain production, 282 million tons. Again, in the coming year, it will increase. But the present situation of the GHI, Global Hunger Index, it is not that happy for the India. Whether it's a nutritional deficiency or it is availability of the food as such to feed the number of the stomachs in the, and this is, I feel it's a very grim situation. Out of 117 countries, it is not. Our neighboring countries, small countries, they are good. But our position is not good. So there now the younger generation should take over, and how to fill this gap, how the rank will come up to the low level, which will be good for the India. That is what there is a big things to work on. Yeah, this is the situation. If you remember the Bengal famine, so the things are goes, the million of people dies. That is what that is the importance of the food. And the philosopher like a Seneca says, the hungry persons listens neither to reason nor religion or nor bent by prayer. So just to experience yourself by keeping yourself hungry for the 5 days, and on the 6th day, if you ask that, okay, you get fresh, put your prayer, and then you eat. You will say you forget about all. First of all, you give me the food. But that needs the experience that you can see also. The challenges, everybody knows that the population, climate, the soil fertility, all these are the big challenges. And the solutions are, first of all, developing the high-yielding varieties. And with the recent trends of the climate says new diseases, the new abiotic stresses, and therefore the adaptation of the variety of climate change. And this is what, again, if you see the nutritional hunger point of view, we have increased our yield. But if you see the quality of the high-yielding varieties or the nutritional composition of the high-yielding varieties compared to our old germplasm or the old varieties, it is not that good. And therefore, I think this particular training program is for that. So that is the major challenges. Now, what has happened so far, it is up to 218 or 19. This was okay. We are getting the food. We are producing the high-yielding varieties. We are managing the population also. So from productivity to the getting the food by each and every person, it was okay. But now onwards, this gap has started, and it will increase in the. This is going to be food demand, and this is what the food supply in the future. So therefore, increasing the productivity, it's a per unit area that will be the major challenge by avoiding the loss and avoiding the food supply. This is what the major challenge. So this should be narrowed down in the future. And therefore, our developing the high yielding varieties with the better nutritional quality will be the major challenges for all of you and all of us. So far, we were doing the conventional plant breeding, and we have reached to this stage, including some of the biotechnological aspects. Okay, but now the things like various omics technologies with the it's like CRISPR and tilling is going to play a major role for the modification of the crop plants. But beside this traditional mutation breeding and the omics or the biotechnological aspects, I emphasize on the mutation breeding. You ask me why? Why the mutation breeding? Now, suppose if you take any crop, say like a wheat, rice, or in that case any pulses or any oil seeds, you find that the yield potential of all these crops has reached to the particular stage. Say like a wheat, suppose you are getting 60 quintals per hectare. With our efforts, it will go to the 70 quintals per hectare, 80 quintals per hectare, and now we are expecting 100 quintals per hectare. Okay? Say like rice also. So I was in the BISA some 2 years before, and they put all varieties in a row. The released varieties since 1950s to 1918. And I found there is hardly any difference. Okay, hardly any difference. But still, every year we release the variety showing that it's a 5% increase, 10% increase, and all these things. Now, suppose I have the plant which is giving the, say, 70 quintals in a farmer's field in that case. Okay? Our experimental may go up to the 100 quintals. Now, above this, suppose you want a yield increase. What is the solution? The first solution, unless and until you have the variability for the particular character or the range of the character, which so far in the last 50 years, people have exploited or the scientists have exploited the all germplasm, you have done all possible crosses and come to the stage of the 70 quintals per hectare. Now suppose if you want to go beyond this, unless and until you have the variability for the particular trait, the challenges which I put it over there for that particular trace, yield and yield contributing character, resistant to biotic and abiotic stresses, novelty in the nutritional character, then and then there is a possibility to go ahead. Otherwise, using the two high yielding varieties, we have the experience, there is hardly chance of increasing the yield potential. Suppose there is a one variety of 70 quintals per hectare, another is a 75 quintals per hectare. I cross these varieties and try to isolate a recombinant which will be the 80 quintals per hectare. The chances is very less because I have put all the possible good genes in the one variety. Therefore, it is giving the 70 quintals per hectare. Another variety which is giving the 75 quintal per hectare, there also I tried to put all possible good genes. It means that they have come to the saturation level. In the genomics point of view or the gene point of view, when all the things are better, so when you cross it, what is the chance of getting the recombinants which will be 80 quintals per hectare? It's a very less, very less. So unless and until you modify the genome, say maybe the biochemical pathway, maybe the physiological pathway or the anything which will try to provide you the new kind of the recombinants, then and then. I will give some of the examples how the mutations have broken the yield barrier of the particular crop. And therefore, unless and until we have the new germplasm in our hand, it's very difficult, though we'll claim that we want to develop a variety which will be 100 quintals per hectare. In my opinion, it will be very, 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 very difficult, unless and until you generate the new germplasm. Or else, you try, if you don't want to do it, then you exploit the all germplasm available in the world, which people have done. Our seniors or those who have worked for the crop improvement, they have slogged their life to develop the high yielding varieties by using the existing high yielding germplasm. And therefore, so there is now, we should go ahead with the new ideas. New ideas is the, unless and until you have the new germplasm in your hand, it will be very, very difficult. Okay? So therefore, I feel the changing the germplasm or isolating the new germplasm, mutations is the only solutions. There is a possibility of, another possibility of using the wild germplasm. But when you use the wild germplasm in the crossing program, you will find a large number of the genetic drags. Unwanted genes comes in there. And to delete that, again it is we have seen in the brassica, Dr. Banga has done a lot of work on that. Even IRI has done a lot of work on this. But in this process, what happens, we have developed the plant, which will have the particular character, but it is not showing the high yielding potentiality, and sometimes the undesirable characters are also getting involved. Now to delete this, again, we'll have to do the mutation. Okay? Unless and until you delete that, and that is again the long process. So in the mutation breeding, what happens? You take a high yielding varieties, okay? Okay, before that, I will see, this is the general applications of the nuclear energy you can do for the pest control. This is our main objectives: food and environmental protection, soil, water, and animal protection. If you really want to go for the details of the application of the nuclear techniques in the agriculture, this. Now this is what I was talking about, that there is a need to strengthen the germplasm. Now how the crop evolved? If you see the evolution of the crop plants, say maize, it was a teosinte like this, and now we have the maize, it is like this. It is nothing but the mutation. The mutation and the recombinations. Now just to think of the maize crop looking like this, and now the big crop like this, and just to say within the hybridization program would have taken a long years too, because there was the mutations, good mutations, and that we used in the crossing program. And likewise, even in the rice or the wheat or even the tomato, if you see, even the sunflower. So if you look in the history of the evolution of all the crop plants, they are the product of the mutations. They are the product. And the very well examples which I told in the wheat and the rice. So without mutations, it's very, very difficult to go ahead. Now why the mutations again? So this is what the spontaneous mutation, their frequency is in the 1 lakh or the 10 lakhs you get it. But in the induced mutations, that frequency is reduced. You can isolate a 1 mutant out of 100 plants. You can isolate 1 mutant out of 1,000 plants or the 10,000 plants. Okay? That is the beauty of the. Suppose I have taken a high yielding variety, and as per my objective, suppose I want to do the better nutritional quality, suppose I wanted to do the resistance to the biotic and abiotic stresses. So suppose I have taken a variety, say SD2967, which is very famous in the wheat cultivation. Around 60% area of the wheat is under that variety. But now suppose if it is suffering from the yellow rust, what is the solution? So that example I will give it to you. So, and generally there is a confusion with the people that whether it's a GMO or not. GMO is a totally different thing. This is manipulation within the genome. It is within the genome. It is not any gene, it's not brought from the outside. So definitely it is. But sometimes people misunderstand things. go back to the little bit of history of the mutation breeding. It's a Hugo de Vries who has coined the term mutation. And the Muller is one person who worked on the Drosophila in 1927, and he got the various mutations, and he got a Nobel Prize for that. And in the crop plant, Stadler is one person who used the X-rays in the barley, maize, and wheat, and from that point onwards, the mutation breeding started. Now, this is just a simple scheme I will tell you. This is the wheat variety with this genome, and we have done the irradiation, and this is how this new gene has appeared, or the modified the genome and the new character has appeared. You get a various mutations. Once you start the mutation breeding, you get the unwanted mutations also. Generally, because the mutations are recessive, and therefore they are the sometimes lethal and the undesirable. So you will have to reject such mutations which are not good, and it may lead to the because the frequency is very, very less. So most of the plants will be like a normal type. So that also you reject. And as per your objective, whether it's a high yielding, disease resistance, adaptation, or the better nutrition, okay, you select. So two things in the mutation breeding is very, very important. So that is, one is you have to fix your objective, and then do the mutation, and you should have the, just now I was talking with our head of the department, and she also expressed that screening technique. Screening technique is a very, 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 very important. I will show you in the next topic. So to start with the mutation breeding, what is required is a lethal dose 50. Fixing the lethal dose 50 is very, very important. How to fix the lethal dose? You treat your seeds with the various doses of the gamma rays. There are the different kind of the mutations, physical and the gamma rays, because generally we go for the gamma ray treatment. So fix the dose of the gamma rays and see how the growth is happening. So somewhere, if there is a reduction, 50% reduction, then the control one, that is called as a lethal dose 50 or the GR50. Lethal dose 50, where suppose you have taken the 100 seeds, irradiated, and where the 50% survival is there, so that is called as a lethal dose 50. So upon fixing that one, the various changes occurs at the genetics level, at the chromosomal level, at the gene level, okay, and in the chromosome level, it is like a normal deletion, deficiencies, duplication, and the inversions, translocations, all this is happen, and the gene level, say like a transition and the transversion, okay, changes within the purine and pyrimidine, between the purine and pyrimidine. So that is how the break happens in the DNA, and you get the large number of the variations. This is what the overall protocol of the mutation breeding. So you start with the variety. Say, suppose I have given the example of the SD2967, which is a highest yielding variety and covering a large area. But suppose it has a defect, say like a yellow rust. Yellow rust is a new disease. So now my objective is to isolate the yellow rust resistance in the SD2967. What is the advantage of it? Already it is a high yielding variety, and I want to rectify the one particular character. Say, now you people will use the CRISPR, but I will go for the mutation. The difference is because you have the gene in your hand, and that you will manipulate with the CRISPR, okay? But the way again the reports are coming, we do not know with the CRISPR again it will be precise or not, okay? I also don't know, because I am doing the hitting my seed in a dark, okay? Bombarding, taking the pistol and hitting in the dark. I do not know whether the mutations happened or not, okay? So what I do, I treat the large number of the seed in the M1 and grow the large population, say like a 50,000, say like a 1 lakh, okay? I will grow this population in the area where there is an endemic situation for the yellow rust. What I said? Screening technique. unless and until you fix the screening technique, whether it's a biochemical analysis, whether it is a field analysis or any character, unless and until you fix that one and standardize your screening technique, then and there is possibility. And in that case, I will say, you dream the character. You select a high-yielding variety which is popular, and you dream the character which you want to modify, but you standardize your screening technique. Your dream will be fulfilled. Your dream will be fulfilled. I will give you some of the examples for that. So, in the M2 population, when you grow the large, in the M2 generation, when you grow the large populations, this is the kind of some segregation you will get. This is just an example for the chlorophyll mutations. This is the normal type green and this is the yellow. Okay? This is the lethal mutations. Likewise, in the Mendelian fashion, suppose a one particular plant, say I have taken the 10,000 seed, irradiated, and I have grown their plant to row progeny in the M1 generation. Okay? 10,000 plants plant in a M1 generation. And when I harvested it and grown the 10,000 plant to row progenies, maybe one of the plant progenies will segregate for the particular character. Okay? So, this type of this. Now, this, because it is a mutation, because it is a random, so I do not know in which plant there is a mutation. So, unless and until I fix my screening technique, say with yellow rust. So, what we have done? We have put this in the BISA campus where we have done the artificial inoculation also, and there is a natural condition also. So, almost all plants, because the original variety is susceptible, they have shown, and there were only 5 to 6 lines which were segregating for the resistance and the susceptible. So, job is done. The population was like a 50,000, 60,000 plants. Okay? So, within a year or within a 2 year, we could isolate that particular mutations. Okay? So, when otherwise in our traditional breeding program, you first of all screen and then you try to transfer that character to the high-yielding varieties with the backcrosses program, and then you develop. Now, within 2 years, we had a mutations. Now, we are evaluating that one. It is, some of the mutants are as good as the SD2967, and some of the mutants are better than that. Some of the mutations are better than that. Okay? Because the mutations, it is not only for that character, though we emphasized, though we fixed our objective for the yellow rust, but because it is a random process, so it has given the mutations for the other characters too. It is hitting in the genome. So, simultaneously, we can improve the large number of the character. But do not dilute your efforts for the other character. You fix the character, first of all, isolate that your mutant, and beside that, because it is a hitting in the genome, you will get the large number of the population. So, you can isolate that also. So, this is how you have to fix your objective, fix the varieties, okay? Standard screening technique you follow and you isolate the mutations. I was talking with Dr. Ranjit now. We have given one project to the BCKV and now in the cotton HTBT is coming, okay? Herbicide tolerance also. So, we thought that why not to go ahead, okay? And in the lentil last year, in the M2 population, the population must be more than 2 lakhs population. And out of that, 2 progenies we got the herbicide tolerance, okay? And one of the very good example in the IRI, Dr. Ashok is there, he has developed the rice. That is also through the mutation. Then he transferred to the high-yielding varieties, okay? So, our generally we say, as I said, that you dream the character, fix the high-yielding varieties and then fix the standard screening technique. There is a likelihood of getting your mutations more than 99%. That is all. And ultimately, this is what the fixation of the mutations, breeding, and then evaluations and the release as variety. Okay? What are the characters? As I said, I have given the example of the disease resistance. You can go for the yield and contributing characters, morphological character, flowering, early flowering nowadays. Okay? The pulse varieties which are coming 60 days with the high yielding potentiality. Okay? Plant type. This is what in the future it is going to come as a one of the perspective, though it is a very old concept of the 1966 which are given by the Donald in the wheat. But now, because of the shrinkage of the resources and the bombarding of the new problems with the climate change, we will have to develop a plant which will sustain under such adverse conditions. So that is what again the major objective. Then resistance to shattering, resistance to biotic and abiotic stresses, seed dormancy, anything, in oil quality or the protein adaptability, biochemical pathways, whatever nutritional. Ma'am was saying that now we should develop a rice variety which will we will eat that carbohydrate, okay, or the starch, but will not have that glycemic index. Okay? So these are the challenges. Now because the people are, now we will have to work accordingly. So in my opinion, mutation is the best option for that. You can go for the CRISPR, you can go for the tilling, you can work for the molecular, but develop first of all that kind of the variability. That is possible through the mutations. Now I will give you some examples. Okay? As I said, you dream the character and you develop the mutations. Now just I will rush through some of the slides. You see the flower color, you see the leaf shape, seed type. This is the brassica again. You see the number of the packed seed. This is what the standard control, which gives the 12 or 13 seeds standard, and here we have near about 21 seeds, but the silica is small. Okay? If you increase the number of the seeds, maintain the number of the silica or increase the number, your yield is likely to increase. Okay? Seed color, if you see, any kind of the seed color, okay? This is what our parent, and we isolated, and we have released the varieties with the collaboration with the PDK via Cola. This is the first variety of the India, Surya. Surya is the sunflower is not the cultivated crop. It was introduced somewhere in the 1960s. But this is what the germplasm we got. Though it was a high yielding, it was not preferred in the market because the oil content was less. The preference is for the black seeded. So maintaining the yield potentially of the Surya, we have isolated a black seeded mutant. See, this is the standard flower and the variability which you got in the sunflower, flower color. This is one of the dwarf mutant which you isolated and characterized, and now possibly in the this year or the next year we are going to put in the yield trial. You see, this is the Varuna high yielding varieties, very highly adapted varieties. Okay? But if you see, this much portion is used in the reproductive phase, but most of this portion it is going as a waste as a energy, though the biomass is good. What we have developed, a dwarf plant, okay, where the vegetative phase is not much, but the yield potentiality of this is as good as a Varuna. If you look into this, there are the two plants, you see the number of the bearings of the silica. So that is how we have to, as I said, tailor the plant type. Okay? Now, with the minimum resources and the agronomic practices, because this is dwarf early, it will exhaust, it will try to utilize maximum nutrients within the short period, and it will give the same yield. Now, this is going to be entered in our All India coordinated trial. This is the early linseed. This is one of the, with the climate change, the farmers are facing the problem. It's our crop is ready, but suddenly suppose there is, even last year I think there is a lot of loss of the wheat, and if this happens when there is no sources for the resistance, for this is the pre-harvest susceptible, if the rain comes, your total wheat is gone. But suppose you develop the mutant, okay, which will be the pre-harvest sprouting resistance. So we have isolated, even in the same mutant population of the ESD 2967, so we have isolated the pre-harvest sprouting resistant also. Okay. This was the example I was giving for the yellow rust. This was tested in the Bisa in the Punjab, and we found that our mutant was resistant. Clear cut. No, I will say the immune type. Sometimes even we say the resistance grade 1, grade 2, though we have that kind of the mutations, but some of the plants were immune, even not a single spot of the yellow rust on the plant. So that kind of the mutations. I was talking about the herbicide. We have conducted the experiment on the herbicide also, sprayed where most of the plants are died. Okay? But we got a few plants which shows and this is to be confirmed. This is just, it is not this is just for the information. We want to confirm this in the subsequent generations. With the drought situation, the level of the water is going down and down. So if you develop the plant which will have the long rooting system, so there is a possibility of the survival of the plant even in the drought situation. So these are the examples with the other crops also. So using this kind of the mutations, I can show you the large number of the pictures, but to cut short the stories, I have put some of the specific examples. So it is not only the BRC or in India we are using the mutation breeding. Mutation breeding is being used all over the world, and using this kind of the technique, so far 3,250 mutant varieties have been developed in the 175 crop species and the 60 countries and the 91 years of the efforts. These are the list of the various countries, how the China is topping, followed by the India, and then the Japan and the various countries. This is what the percent contribution. The maximum use of the mutation breeding has done in our continent, that is Asia. This is again the one specific example I will give at the international level. In the Japan, say 50 years before 60, there was a devastating disease called as a black spot disease in the pears, on the pears. And almost all cultivated varieties, all the plants in the Japan, they suffered a setback of the black spot disease. But during that time, what they found, there was one plant in their gamma field. Okay? There was a gamma field in the IR also, but nowadays it is. So they cultivated the fruit trees and the other crops. The gamma field is a area where they keep the source of the gamma source, okay, cobalt 60 source, and every day they will expose the plants, okay. So when the energy comes or the rays comes, they will hit to the growing buds and there will be the mutations. So during that situation, what happened, there was only one plant of the pears and there was only one branch which was not suffering from the black spot disease, okay. So what they have done, they have isolated that and then they have vegetatively propagated, and the present day cultivation of the pears, now Japan is the one country where the lot of exports of the pears it is out of only that particular branch. There are the two varieties they have produced out of that, okay, which is resistance and present cultivation. So that kind of the miracle can happen this is another miracle, I will say. The world was worried about the UG99 disease in the wheat, okay, which was first appeared in the Uganda and it was likely to spread to the all over country. And there was a big measure was taken even by Indian government. Our germplasm was sent to that area, then they screened and all this thing, and we have now but there was no solution. So what the peoples have done, Miriam Kiriana, and he is a Kenyan plant breeder, he initiated a mutation breeding program. He has taken a 10 kg seed of the 5 varieties. Now just to imagine 10 kg seed of the 5 varieties, the number of the seeds, total number of the seeds. What I was saying, you grow the large M2 population unless and until your M2 population is very, very large, because your frequency is, say, suppose 1 out of 100 or 1 out of 1000 or 1 out of 1 lakh. With that proportion, you have to grow your population and screen under the stringent condition. That is what, again, I want to emphasize, your standard screening technique. Okay? So they have grown that maybe the 5 hectare or more than that area in the M2 population, and then they have isolated a 7 mutant out of that, and now 2 got released. And there were large number of the seminars, symposiums, meetings, policies over the UG99, and the solution came out of the mutation. Okay? So when the doors are closed in the crop improvement program, I will say that start mutation breeding. And these are the examples. It is not because I am working on in the Bhabha Atomic Research Center and therefore I want to emphasize. No. This is the way that when there is no solutions, so only way one can come out through the mutation breeding. Vietnam. Vietnam, once upon a, he was importing the millions of the rice for their survival. And when they started the mutation breeding in 2007, they have started the exporting the rice. That is how examples. So like this, I can give a large number of the examples of the success stories of the Japan, Italy, okay, China. China is the one of the varieties. He is the first to develop the near about 700, 800 high yielding varieties in almost all crops. In the once I visited to their Chinese research station in the rice, they have showed me a plant which was only single come. Okay, at that time first time they reported that was a mutation. So they were doing all molecular biology of that. Through the mutations, there is a solution for the existing problem, and at the same time you can develop the material for the basic studies to understand the plant. Okay? Now once you say the rice, there are a large number of the tillers. Suppose I wanted to study what is the phenomena of the tillers. What are the genes responsible for the tillers? So unless and until I have the contrasting character for that, then and then I will be able. You see the Arabidopsis mutations, why the genomics is successful or why the sequences was successful in the Arabidopsis, because they isolated large number of them. You name any character, they have the mutations for that. So in the genetics or the breeding or in the genomics, unless and until you have the contrasting character, then and then you will initiate the genetic studies. Then and then you will initiate the biochemical studies, characterization or anything. Okay? So beside our big success of the achieving the particular objective, you can generate the large population for the basic studies also. I have developed within the same population of the sunflower, I have developed a plant which was just 10 centimeter tall. Just 10 centimeter. I have not shown the picture of that. Just 10 centimeter. Okay? But I have not done any detailed analysis of that. So what I meant to say, there is a possibility of isolating the large number of the character. And in the USA also, they have done. This is the drought tolerant in the Cuba country, tomato they have developed. So this, you see, this is the first released varieties in India using the different mutagens and the crops. And again, it is classified as per the various crop plants. Okay, the number of the mutants now must be 345 or so. And the character is a high yielding, early maturity, disease resistance, quality, grain quality, abiotic stress, improved plant type, and the other characters. Okay, this I will give the example of IRI itself only. In the chickpea, these are the varieties developed through the mutation breeding, and they were so popular. Their breeder seed production as well as the certified seed production, it was really phenomenal. So this is how the mutations can help even in the increasing. In the Bhabha Atomic Research Centers, we have developed near about 46 high yielding varieties in the oilseeds, pulses and the cereals. I will just go through some of the examples like groundnut. We have done I will say the miracle, our TAG24. It's a drought tolerant variety used in the summer cultivations, and now it is being used as a check variety. So there are the two possibilities. When you have taken the high yielding varieties, inducted the mutations, isolate the mutations, the same mutant can go as a variety, or if it is not giving that high yield, what you can do, use that mutant in the crossbreeding program. So that is called as a cross derivatives of the mutant variety. Okay, they are the two channels. Okay, so our example, some of the varieties are the direct mutant, and if you use in the crossing program, that is called as indirect mutant varieties. Okay, so using both the approach, we have developed this varieties in the various crops. This is a list of the this is the TG37 very bold seeded, 41 and TAG24. It's a way. And if you see we have almost covered all India for this. These are our popular varieties in the various states, almost all over India. Again, our bold seeded varieties. And this is how the farmers are happy in the various states to cultivate our varieties. Now, I will say you can see near about 10 to 11% of the breeder seed indent. Now, I will say in India, there are how many 52 agriculture universities, ICR institutions, and it is a big force. And suppose I say BRC, if there is only one person, or at the most 2 persons working on the particular crop.